Hi, it's Ms. Bastido, and today's topic that we're going to talk about is mutations. We'll talk a little bit about the different types of mutations. When you look at the things that you have to know for this unit, you have to know about gene mutations and chromosome mutations, be able to interpret karyotypes, and then also be able to talk about different mutagens that could cause certain types of mutations. So the definition of a mutation is any kind of permanent change in the DNA of an organism. So this could be from one nucleotide, so the A's, T's, C's, and G's, up to many nucleotides, up to whole parts of chromosomes, which would include probably more than one gene. And so when we categorize mutations, we would categorize them into two main categories. We have gene mutations, and then we have chromosomal mutations. The gene mutations are found, we're talking about specific nucleotides within the DNA. Chromosomal mutations are whole pieces of chromosomes or even whole chromosome mutations. And that usually happens during meiosis that would occur. And so we'll start talking about with gene mutations. We'll start with that one. And there's two types. There's point and frame shift mutations. If you're taking some sketch notes, you really probably, if you made this really big, you probably could just sketch note under each one, and this could be your whole sketch note. For most of the section, you might want a little bit about mutagens as well. So with the gene mutations, one type of gene mutation is a point mutation. So that's just a substitution. And so you can see here, at the DNA level, if the triplet pairs were supposed to be TTC on the DNA and it got changed to TTT or ATC or TTC or TGC, any of those, that you'd end up with, in some cases, no change at all. We call that a silent mutation, because we know that there are more than one codon that codes for an amino acid. And so because of that, we would have no change here. You don't need to know about the silent nonsense and missense, but it might be useful just to be able to think about that. If you get a problem on an exam or something and it didn't cause a change, you might be confused. So just know that that is possible. If this letter was changed, it was changed to a stop codon. We call that a nonsense. That causes a huge change and a much bigger change than just, or potentially a much bigger change than just one amino acid changing. And then these are missense mutations. These would really depend on how bad they were, would depend on what was being changed, what the amino acid was. So if this amino acid was hydrophobic, and this one was hydrophobic, it might be okay. But if this one was hydrophobic and it got changed to a hydrophilic, then that could cause a big change in the folding of the protein, which would then cause big changes. And so they have an example of that, like sickle cell anemia, which is caused by a point mutation. So that's an example of a small change, but has a huge impact. And so the other type of gene mutation is a frame shift mutation. And in this case, we either have an insertion or a deletion. So a nitrogen base has been inserted where it doesn't need to be, or one has been deleted. So in this case, I think one is being inserted. It leads to an early stop codon. That might not always be true, but basically after that insertion point, every amino acid after that would be different. And so these frame shift mutations would always have big consequences. So you can see here in this case, this is a mutated DNA strand. We've added an adenine. Now we have a mutated mRNA, and we have these two leucines here, which leads to an incorrect amino acid sequence. And you can see that again here. So it could also be that you're taking one away, and so that's also, that's what they're doing here. Sorry, they've taken away the adenine, and that leads to leucine and alanine. The other one led to leucine and leucine. So both of them were mutated, but mutated in a different way. Chromosomal mutations, there's a lot more different types. We could have a deletion, a translocation, a duplication, or an inversion. So you can see here in a deletion, part of the whole chromosome has been cut out and deleted. It could be like at the top. This one, they're showing one in the middle. But a piece of the chromosome is missing. So potentially, if there's a lot of important genes on here, this could be catastrophic. In a translocation, a segment or a piece of the chromosome is transferred to another chromosome. So put onto the other chromosome. It could even be its homologous pair, but a piece of it breaks off and goes onto the other chromosome. Again, that could potentially be catastrophic. A duplication is a segment of a chromosome or a piece of chromosome that's transferred to its homologous pair. So it's duplicated, being that it's the same exact sequence again, or the same exact genes repeated twice. But this chromosome here would then be missing them. So in this case, it's in the translocation, I think I misstated, but the translocation, it would be transferred to another chromosome that is not a homologous chromosome. And the duplication, it would be transferred to the homologous chromosome. And this would happen during crossing over. So essentially, this one tried to cross over, but this one didn't break off and transfer. And so you end up with this duplication. Also, an inversion is possible. And that is when a chromosome is, a piece of it comes off, kind of like the deletion, but then it flips around, rotates 180, and then reattaches. So now the genes are in the wrong order, which also can cause issues. So when we look at some examples of these, an example of a deletion is Cri-du-chat or cat-cry syndrome. And so in this case, chromosome 5, a portion of chromosome 5 is deleted. This is a pretty serious syndrome. That the reason it's called this is because babies that have it typically, when they cry, sound like a cat. And so that's where it gets that name. The last type of, and probably the most important type that you need to know about of chromosomal mutation is nondisjunction. So nondisjunction occurs not when chromosomes are crossing over, but instead when chromosomes are supposed to separate during meiosis. So it could happen in anaphase 1, or it could potentially happen in anaphase 2 when sister chromatids are supposed to separate. So in this case here, you see homologous chromosomes are lined up and they're supposed to be pulling apart to opposite ends during anaphase 1, and that's what's happening here in B. And that is what's happening in G correctly. But in B, both homologous pairs go to one side. And because of that, we end up with this extra chromosome here, and you'll see that in E, we have 3 chromosomes in each cell, and then in F we have 1. And so we were supposed to be halving the number of chromosomes, so we should have, the only one here that's correct is L, where we have 2 chromosomes, which is what we were supposed to end up with. And so we now have, in this case, in H, we had nondisjunction occur in anaphase 2. So you had nondisjunction occur in B, where the 2 homologous chromosomes pulled to the same side. In H, we see that the sister chromatids didn't pull apart. And so we have lots of mutated sperm or egg cells here where we have extra or missing chromosomes. If you have an abnormal number of chromosomes, we call that aneuploidy. And so this is going to affect every chromosome or every cell in the body of the developing fetus once this egg or sperm is united with its opposite. So if we look at a karyotype of this, a karyotype is a picture of someone's chromosomes. And then this picture is then put together to show, we've analyzed these in class before, but we pair up the chromosomes. In humans, chromosomes are numbered 1 to 22, and then there's a 23rd pair, that's the sex chromosomes. And so in this case, we have, you can see in this case we have, you should have 2 of every chromosome. And so in this case, we do indeed have that. And because there's a presence of a Y chromosome, this individual was assigned male at birth. And so this would be a normal karyotype because there's 46 chromosomes. So if we look at some genetic disorders, this is kind of not a real picture of chromosomes, but you can get the idea. So this is Cri-du-chat, and you can see here at chromosome 5, a portion of it has been deleted. And again, this is a male because there's a Y chromosome there. And you know it's a Y chromosome because it's a lot smaller than that X. With Klinefelter syndrome, what happens is during meiosis, 2 X chromosomes end up in 1 egg or sperm. And so we have an individual that is XXY. So this would have likely happened in the female, but in the mom, but it could happen in the father as well, although it seems to be more common to happen in females in the formation of eggs. And so here we have an individual that aneuploidy has occurred. There's 47 chromosomes with an extra X. Because there's a presence of a Y, they'd still be assigned male at birth. And some males have no symptoms, but a lot of them do. And so they have varying degrees of cognitive, social, behavioral, and learning difficulties. They usually have a decreased level of testosterone, and they might even have inability to not have children. So this is called Klinefelter syndrome, an example of aneuploidy. Another example of aneuploidy is Turner syndrome. And in Turner syndrome, instead of having 47, which is an extra chromosome, we're actually missing a chromosome. And so the egg or the sperm had an X chromosome in it, but the opposite of that, the egg or the sperm, did not have a sex chromosome in it. And so let's say the egg did not have a sex chromosome in it, but it was fertilized by a sperm that had an X, then we would end up with Turner syndrome. So this is also an example of aneuploidy, and but with 45 chromosomes instead. Typically, we have lots of symptoms here. Usually, the ovaries don't really produce viable eggs. Usually they're short in stature. They have like a fold of skin around their neck and swelling of the hands and feet. So this is another example of aneuploidy. An example that you're probably more familiar with would be Down syndrome, which is the most common type of aneuploidy. And in this case, we also have an extra chromosome, but it's number 21. And in number 21, there's an extra chromosome. And so this individual, somebody with Down syndrome, has 47 chromosomes instead of 46. And typically people with Down syndrome have a lot of the same characteristics and have some commonalities among them. So they have almond-shaped eyes, they have flattened faces, short necks, small ears. Usually their tongue sticks out a bit. And occasionally they'll have some other health issues involving their heart. But not all of them do, so that kind of varies from person to person. So all of these are examples of aneuploidy. There are other examples, but then there's examples where this would never happen. So in chromosome 1, for instance, there's too many important genes on there. You really can't have an extra or missing chromosome in chromosome 1. So there's chromosomes where it happens often or semi-often, such as Down syndrome. And then the other ones, even the few other ones that it's possible in, it doesn't happen very often. So there's different ways in which we can end up with mutations, specifically gene mutations. We can inherit those. So we can inherit mutations from our parents. And this is actually really important because some mutations are advantageous, and that's what drives natural selection. And so not all mutations are bad. And another way we can get mutations is we can acquire them within our lifetime in our non-reproductive cells. So if it doesn't happen in my eggs, then it's not going to be passed on to my offspring. And so an example of that is if I were to get skin cancer, that skin cancer doesn't get passed on to my offspring. All cancers would be examples of this, but there are other examples, and we'll talk about those in a minute. So a mutagen is any agent or chemical, it could be a chemical, it could be radiation, that causes a genetic change in the DNA. And so we know that UV radiation causes that. We know that X-rays cause that. Often things that you think of that are carcinogens, which are things that cause cancer, are also mutagens, like those are one and the same. So, but there can be things that are mutagens that are not carcinogens. So something else to think about. So cigarette smoke, usually nitrates in preservatives, benzoyl peroxide, which is sometimes found in acne products. The HPV virus is an example of something that causes mutations. And then also this Helicobacter pylori bacteria, which can be found in contaminated food can also cause mutations. So if we go through each of these, I'm not going to spend too much time on them, but just giving example, any kind of physical mutagens or anything like ionizing radiation, X-rays, radioactive decay, so like from a power plant, and solar radiation, any of those can cause changes in the DNA, and those changes can lead to cancer in most cases, but they could also cause some other problems as well. Chemical mutagens, these are really mostly unknown. There are some that are very well known, such as asbestos causing cancers of the lungs and the lining of the heart. Boric acid is an insecticide. Nicotine found in tobacco, nitrous acid, sodium bisulfite. All of these are known to have some level of mutagenic behavior, but we really there are lots of chemicals out there that we wouldn't know, and so just being able to kind of understand that there are chemical mutagens. There's no sort of expectation that you know like a laundry list of the certain ones, but I just put a few examples here. Biological agents can also cause mutations, such as viruses. This is probably one of the most common ones that most of you have probably been vaccinated for by now, and so this can cause cervical cancer. So the HPV virus is sexually transmitted, and there is a vaccine for it. So it doesn't always cause cervical cancer, but it puts you at a much higher risk for having cervical cancer, and so important to be vaccinated for that. And that is about all over mutations. If you have any questions, Omar is here to answer your questions. That's actually a very young picture of him. He's much bigger now. I hope this video helped, and let me know if you have any questions.